Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I'm Dr. Demi, as you already know if you've been following my channel for a very long time now. And in this video, what I want to do is just show you some of the changes that have been made to the syllabus for the 2022 to 2024 class. So in case you're unaware, the biology syllabus, or actually the CIE syllabi, change about every 3 years. And the changes are not always life-shattering. So if you've been studying and you're going into 2022 to write your exams, please don't be afraid. Some of the things are things that you just need to brush up on, and they're not very hectic changes. So what I've done with this video is I've just prepared snapshots of the different changes that have been made, and I've also included a link in the description for where you can find the syllabus so that you yourself can go through it. And just want to say that when I do teach in my class, my students get the syllabus for each topic that we're covering. They'll get what the syllabus says, and while we're teaching or learning, they can then tick off as they learn and say, okay, this part of the syllabus has been fulfilled and I need to maybe brush up on this part. Obviously this was before COVID, so the students I had during the COVID era did not enjoy that benefit, but by all means employ it as a means to see what you're doing right and what you still need to brush up on, and what you might need to independently study in case your teacher has skipped some of the things that you should know for the 2022 exams, or in case you just haven't had the privilege to learn those things because you haven't been able to go to school because of COVID. Okay, let's get into it. So the first things first, I'm just going to get my red pen here as usual. First things first that was very evident was the changes to the times, the time allocations for the different papers. So you already know that if you're writing the A-level syllabus, so if you're writing the whole syllabus, you have 5 different papers. Paper 1 is multiple choice and it's usually for 1 hour, but this was the old syllabus over here. So I'm just going to write old there. And in the new syllabus, the time allocation for paper 1 now has an extra 15 minutes. I still encourage students to try as much as possible to answer the 40 questions within 1 hour, and try to use those 15 minutes if you're in the exam to sort of go back over your answers in case you might have wrongly selected something or you need some more time to think about a particular question. The weightings for the different papers are still the same. As you can see, paper 1, if you're writing only at AS level over there, it weighs 31% of your grade, and it's still the same in the new syllabus. And if you're writing the full syllabus, then it weighs 15.5%. Paper 2 has also had an additional, it hasn't had additional time. It's still the same, 75 minutes, just as it was in the old syllabus. Paper 4 is also still 2 hours, just as it has always been, and the same with paper 5. And paper 3 also has 2 hours as usual. Also remember, if you're writing paper 3, that when they say you have 2 hours, it doesn't necessarily mean that you have 2 full hours to just go on with, like you probably can't shoot over time with some questions, so you have 1 hour per question basically. So it's very important to bear that in mind and not say to yourself, oh well, I think question 2 is a lot easier, so I can spend some extra time on question 1. Some centers would not allow that, so please make sure that you are on top of your game and you're able to complete in an hour. So this is a good time if you're writing in 2022 to start to get your teacher to give you practice sessions in terms of the practicals, so that you can become really good at them and you can understand all the things that need to go into completing a practical in an hour. Okay, this again is just showing you the different routes that you can take for this syllabus. So you can decide to write at AS level only, which means you would only write papers 1, 2, and 3. And these cover chapters 1 to 11, and this is like an experimental paper. So paper 3 is a lab paper. If you're writing at A level, that's over 2 years. In your first year, you write your first 3 papers, which again is the same as the AS content. And in the second year, you write papers 4 and 5. This is very dependent on the school where you're studying. Where I used to work, some of the students actually don't write any paper at the end of the first year. They only write all 5 papers at the end of the second year. And that is this third track over here. So that's the one that they follow. So it means that over 2 years, they are sort of prepared for all 5 papers and they write everything at one go. All right, so now just looking at the content overview of the new syllabus, everything is pretty much the same. Maybe some names have changed, but it's more or less the same content that you would be covering. So if I were you, I would not be too stressed about it, although there are some tiny little details, like I said, that have been changed that you might need to pay extra attention to. And again, you might say, well, I mean, this is something that I haven't been told. Well, you're being told now. So please, by all means, make use of the information that you're getting on this channel so that you can boost your grades and also improve your performance in school. Okay, so what I've done is basically taken the chapters that have changes and sort of just shown what the old syllabus said and what the new syllabus is saying so that you can have an idea of what has changed exactly. So in the old syllabus here, it says that you need to compare the structure of a typical animal and plant cell. But over here, when you look at what is requested of you, you're not actually expected to do that comparison. However, it is very difficult for you to study this chapter without being able to compare the two of them. So I don't think that is a significant change, but I just thought to highlight it that when you do get into the exam, chances that you would be asked to do this comparison are very slim. And this is for the exam in 2022. For those who are writing in October, November 2021, you are still working with the old syllabus. So please, by all means, stick to that. And don't try to incorporate things from the new syllabus because your work doesn't cover that. Here in the old syllabus, this is for chapter 2, biological molecules, where you learn about glucose and all of those things. And if you haven't checked out the channels, the videos on all the different topics, please by all means do so because they're very helpful. Most of the students I know who have used them and written their exams said they found them really, really helpful. And what many of them would do is just watch the video and then go and practice past papers with that knowledge, and that really improved their grades. So please check them out. But when you do chapter 2, normally in the old syllabus, students are often able to describe the ring forms of alpha and beta glucose, but now that has been explicitly said that you should also be able to draw these ring forms. So you should be able to tell the difference when you're drawing between an alpha glucose and a beta glucose. Something else that was also interesting here is that you're expected to know the role of covalent bonds in joining smaller molecules together to form polymers. So that might be something that you haven't paid much attention to, but now is a good time to just read up on it. And again, just take note of the word state. So state means that you don't have to provide too much detail. It's more like just stating in very brief sentences or in an outline form even what you're trying to say. Okay, in question 3 here, this is something interesting. This wasn't included before. So before, you can see here that you can investigate the progress of an enzyme catalyzed reaction by measuring the rates of formation of products. But here it's very clearly saying that you should be able to outline the use of a colorimeter for measuring the progress of enzyme catalyzed reactions that involve color changes. So this is something you need to be able to do, and it says outline, so it's not asking you to necessarily describe an experiment. So it's just saying state how a colorimeter would be able to do this. So this is something that you might want to read up on or just follow up with your teacher so that you are better prepared. Here in 4, for some reason I don't know why I didn't circle it, but I believe here is the main thing here. So if you look at C over here, it does say that you should outline the process of cell signaling involving the release of chemicals that combine with cell surface receptors on target cells, leading to specific responses. But here it's making it very clear that you should be able to outline the main stages in the process of cell signaling. Again, if you are a very diligent student and you are very detail oriented, by doing this you would have already fulfilled that. But just I put it in here just so that you can see that it's now being specific by saying you should be able to speak about the secretion of ligands from cells, the transport of ligands to target cells, as well as the binding of ligands to target cells. So just that means that when they speak about when they ask you a question on this, they expect you to speak within these 3 phases that have been identified. Something really good about the syllabus as well is that when you look at the syllabus, you can actually tell that some of the things that might be questions in the exam have already been provided for you. So I always encourage students to get a copy of the syllabus. For my students on Canvas, what we do at the beginning of every academic year is the syllabus for that year is uploaded so that they can see it and they can access it at any time on Canvas, and that way they're just able to walk through it. Okay, so here, this was an interesting one. This is for chapter 6. The changes in chapter 5 were not that deep, so I just did not include chapter 5, but I've put chapter 6 here. And what I want you to look at here is this big red circle that I've drawn, because you would not find that in the old syllabus. So it says here, describe the structure of a DNA molecule as a double helix, including, and then it goes on to state all of these other things that you have to include, such as the importance of complementary base pairing between the 5' to 3' strand and between the 3' to 5' strand, and also ensuring that you mention that they are anti-parallel strands. Also, the difference in hydrogen bonding between cytosine and guanine and adenine and thymine. And then linking of the nucleotides by phosphoester bonds. That's also phosphodiester bonds rather. That's also something that you are expected to include when you are describing the structure of a DNA molecule. So if this was a 6-mark question, you would be expected to go into all of these details, for example. Then it says that you should describe the semi-conservative replication of DNA during the S phase of the cell cycle. If you look at the old syllabus over here, you can see that that is also included here. But under the new syllabus, or in the new syllabus, this has now been more specific. So they say you have to outline the roles of DNA polymerase and DNA ligase. And look at this. It says now, knowledge of other enzymes in DNA replication in cells and different types of DNA polymerase is not expected. So typically when we speak about the DNA replication process, we tend to go into a lot of enzymes and what they do, topoisomerase and all of those things. But when you are doing the new syllabus for 2022, please note that the key enzymes that you should mention are DNA polymerase and DNA ligase. That doesn't mean that you would get penalized for mentioning the other enzymes, but it just means that the mark scheme might be looking for something else. You should also make sure to mention the difference between the leading strand and the lagging strand, and that would also play a big role for you as you answer this question. Then something that I didn't see in the old syllabus that happens to be here is that you need to describe the structure of RNA, but using the example of an mRNA. So over here, it does say describe the structure of RNA, but doesn't necessarily specify that you need to use mRNA. So that's just something that I'm pointing out to you now. Oh yes, and in chapter 6, it does continue. So it seems like there were quite a lot of minor changes. Again, these changes are really minor. If you're a diligent student and you've been using the old syllabus, it's not going to be a train smash for you. But just bear in mind that over here, you should be able to explain that the RNA molecule formed from transcription is modified by the removal of non-coding sequences called introns and the joining together of coding sequences called exons to form the mRNA. That's not something that used to be in the old syllabus, so by all means pay attention to that. And then also to explain that a gene mutation is a result of substitution or deletion or insertion of nucleotides in DNA, and outline how each of these types of mutation may affect the polypeptide produced. That's very different if you look on the left from what you see here. Because here it just says state that a gene mutation is a change in the sequence of nucleotides that may result in an altered polypeptide. But in this case, you have to explain. So you're not stating. Stating here is just more like defining. But here you're explaining in detail that a gene mutation requires or involves rather all of these things: substitution, deletion, insertion, and then you also explain how each one would affect. So please pay attention to that when you're studying for the exams. In chapter 8, so over here, this actually has always been part of the old syllabus, where it talks about the chloride shift, but not much attention, or should I say not many questions have been brought up regarding this. But when I started looking at the outcomes and I saw it as like the second outcome, I started to wonder if it has become more important. So I would say please have a look at that in case you've missed it, because you've been looking at the old syllabus. So describe the chloride shift and explain the importance of the chloride shift, obviously with respect to transport in animals. But also it goes deeper to then say that you should be able to describe the role of plasma in the transport of CO2. That's not something that was in the old syllabus. So this is something for you to pay attention to if you are preparing for 2022. Also describe and explain the oxygen dissociation curve of adult hemoglobin. So if you've been following, you probably have heard about the Bohr shift. You can also look at the videos on that, but basically just trying to ask you to explain what happens with oxygen as it binds to hemoglobin. So the fact that when one oxygen binds, it sort of contorts the shape of hemoglobin and makes it more susceptible to oxygen binding, which then leads to its saturation with time. So that's something that you would be able to explain. But then that's very different from the Bohr shift, because when you're discussing the Bohr shift, you are discussing the impact of CO2 partial pressure on the oxygen dissociation curve. So the oxygen dissociation curve is a standalone thing if you're not asked to discuss the Bohr effect, but the Bohr effect does bring in the impact of CO2. So please pay attention to that as well. Explain the importance of oxygen dissociation curve at partial pressures of oxygen in the lungs and in respiring tissues. Again, this was not specified in the old syllabus, but you should be able to highlight how the oxygen dissociation curve would be different if the hemoglobin is in the lungs compared to if it's in respiring tissues in other parts of the body. So that's also something to bear in mind. And I will try to do like video updates if I can, just to make sure I cover some of these differences that have now arisen from the new syllabus. But please do the work as well. Be committed to it and sort of search out things for yourself, and so that you can be up to standard with regards to the syllabus. Okay, so here again, I think I've made the mistake of not circling, but one thing that does stand out here that I now remember is that in the old syllabus, you would have been asked, as you can see at the bottom here, to describe the effects of tar and carcinogens in tobacco smoke on the gas exchange system. And you also speak about diseases of the lungs and all of those things. Those are no longer in the new syllabus, so you're no longer required to learn that for 2022, which I guess for some students might be a bit disappointing, because then it makes this chapter, I guess, less interesting for them. But still, you can still study them just to have the knowledge. But based on what the syllabus says, all you have to do is sort of learn about the gas exchange system and how it works, and also be able to describe gas exchange in the alveoli, between air in the alveoli and the blood in the capillaries. And so just also being able to recognize the different parts of the gas exchange system, even if you're looking at them through a microscope or looking at a photomicrograph, you should be able to identify them. In chapter 11, things are also a little bit different. So over here, it speaks about, this is the old syllabus on the left, so it just speaks about phagocytes and so on. But if you look at what I have circled here, now they're asking you to explain what is meant by an antigen and state the difference between self-antigens and non-self-antigens. Those are actually here. But here it's a different story, because it says explain the meaning of the term immune response, making reference to the terms antigen self and non-self. But here in this case, you are explaining what the antigen is and not necessarily explaining the immune response per se. But you can say that if a non-self antigen invades the body, then it can trigger an immune response. And then over here, describe the sequence of events that occur during a primary immune response with reference to the roles of the macrophages, the B lymphocytes, and the T lymphocytes. And what this is simply telling you to do is to sort of write out a timeline of what happens during an immune response, especially when they say a primary immune response. That means the first time the body is being exposed to that antigen, or the first time that antigen is invading the body, whichever way you want to see it. So be able to describe what the first line of defense is, what follows after that, and basically what's the end of that as well. Then here, explain the role of memory cells in long-term immunity. This is actually part of the old syllabus as well, but I guess I circled it out of error, but also just pay attention to that. In chapter 12, there were no hectic differences, so I just moved on to chapter 13. And in chapter 13, over here, you can see I didn't put the old syllabus because it didn't have the information that is now here in the new one. And what you can see here is that you're now required to sort of identify the events that happen in cyclic phosphorylation as well as in non-cyclic phosphorylation. In the past, many of the questions on photosynthesis have not really paid attention to phosphorylation as such. They're usually questions where you have to analyze data, draw conclusions, and maybe state some facts about photosynthesis. But in this case now, you're being asked to explain cyclic and non-cyclic phosphorylation. So if you haven't watched that video, please make sure that you do so. And you can see here that they've even sort of outlined what happens for you, that there's only one photosystem in this one. There's photoactivation of chlorophyll and ATP synthesized, whereas in non-cyclic there are two photosystems: photoactivation, oxygen evolving complex, and then the production of ATP and reduced NADP. All right, again, more errors on my part, but I think I know why. This is actually just talking about blood glucose control in homeostasis. Now, usually you can see here that it has the role of cyclic AMP in the old syllabus, and also just sort of goes into a bit more detail. But here it's going into much more detail by saying, oof, that was a bad drawing. I'm just going to erase that so I don't confuse you. But here, what you can see here, obviously, is that it is asking you to describe the principles of cell signaling. Now, cell signaling, if you remember correctly, is something that you learn about in chapter 4. But when it comes to the cell signaling where glucagon or glucose control is concerned, the specific, or should I say the sequence of events, is really specific. So here what they're just basically telling you to do is that you need to be able to describe the principles of cell signaling and use glucagon as an example. And here they even tell you what would be expected, that there's a hormone that binds, there's an activation of a G protein, the second messenger is cyclic AMP, and then there's an enzyme cascade by the activation of kinase, and that goes on, and they sort of just tell you what the outline is. And this is one of the things I love about the syllabus, that sometimes things you might be too lazy to go into, you might find some of the information in the syllabus, and so when you're covering up later on, maybe studying and going a bit in depth, you have that foundational understanding. So please, by all means, this is one to pay attention to as well. In coordination and control, which is chapter 15. Now, in the old syllabus, one of the parts, and I think this was a pain part for many students, is that a pain point rather for many students, is that you had to explain the impact of hormones like the follicle stimulating hormone, the luteinizing hormone, estrogen and progesterone, and you had to explain how these affect the menstrual cycle and the ovaries and the uterus and all of those things. And you also had to explain contraceptive pills and how they work. In the new syllabus, there is nothing about that. And you can please check in case I might be wrong. Please, if I've made any errors, by all means, post it in the comments so that everyone can see it. Because I don't want you to just take my word for it. And if you spot any errors in what I've sort of analyzed so far, please post that, because I think it would be necessary for you to have the right information information than for me to appear to be right. But from what I did during just scanning through the syllabus, there was nothing on this aspect, and that means that this chapter is now going to be maybe a little bit shorter, and you would not have to maybe deal with things like spermatogenesis, oogenesis, and all of those things. Rather, you would be focusing on how membranes are depolarized, what leads to an action potential, resting potential, and also muscle contractions. Chapter 16 is about inheritance, and over here there isn't much actually. There aren't a lot of differences rather between the old and the new syllabus. But one thing that was quite evident when I was looking at this syllabus was I kept seeing this Punnett squares. And in case you don't know, the Punnett squares are the ones where you do something like that. So let's say we're crossing someone with a genotype of AS with someone who has another genotype of AS. This would then be a Punnett square, right? So you draw them like that, and then you write the crosses. So A times A. If this is a bit confusing to you, please watch chapter 16 on my channel. Just go to the playlist and select chapter 16, and you'd be able to see all the videos on chapter 16 there, so that you can understand what I'm doing. But this is more or less what a Punnett square looks like, and I saw that it was sort of emphasized in this new syllabus. So I'm guessing that when you are writing an exam and you're asked to predict crosses or to show crosses or things like that, you would be expected to use a Punnett square more than anything else. So by all means, please try to use the Punnett square. Obviously there are other ways that you can do this, so I can erase this over here. And there's another way to do this, which would also still be a genetic diagram, but I just picked up on the frequency of Punnett squares, and I'm guessing Punnett squares are a lot less confusing. I mean, even right now I'm already making errors because my mind was just elsewhere. So this is AS and this is AS, so those are the two guys we crossed, and this would be a genetic diagram, right, where you write AA over here, and you then write AS over there. So obviously, as you can guess, a Punnett square might be a lot neater in the exam, and also just show clearly what crosses you've made, because I imagine when students are nervous sometimes they can draw the same thing like 4 or 5 times, erase them, the paper looks a bit dirty and things are just not as clear. So just use a Punnett square, it's emphasized, so try to maybe use a Punnett square. So for chapter 17 and 18, things pretty much stay the same. So you still have to learn about the domains, the kingdoms. And I know that for many of you, you're like, oh my God, why is that the one thing that stays the same? Oh, well, can't do anything about it. But those are not boring, and they're also not hard, because the things that you learn in the domains, like when you're learning about eukarya, or you're learning about archaea, or you're learning about whatever it may be, those are things you actually learned in chapter 1. So I always tell students, like, instead of getting frustrated with what this looks like and how much information it is, just think of it as the things that you learned in chapter 1 when you were learning the differences between eukaryotes and prokaryotes and viruses, and that makes it a whole lot easier. So it feels like revision. Obviously, there are all those other things such as selection, speciation, and all of that, but don't worry, I've got you covered, because all of those are on this channel. So please just make sure you check them out. Then for chapter 19, which is the final chapter, things have changed a little bit. In the old syllabus, you were expected to know the definition of bioinformatics and sort of know what the role of bioinformatics is. That no longer exists in the new syllabus. It's actually been totally scrapped. But what you do have to know are things like the advantages of recombinant human proteins to treat diseases, the advantages of genetic screening for things like breast cancer, Huntington's disease, and so on. And also just showing how you can treat genetic diseases with gene therapy, and also the social considerations. So these are all things that are also sort of in the old syllabus, but in the new syllabus they take center stage, and there's nothing about bioinformatics in the new syllabus. So I hope that you find this helpful. This is where the video ends. I hope that you've found this very informative, and I hope that it sort of inspired you to go look at the syllabus yourself. As a student, I just want to say that it is important for you to not be in the dark regarding this. Please don't solely rely on your teachers. I'm sure your teachers are trying their best and doing all they can to prepare you for the exams, but you also need to take some responsibility for your success. And so I encourage students, by all means, to always have the syllabus handy, and as you cover every topic with your teacher, make sure you are ticking off what you're learning. So if the syllabus states that respiration is a 4-stage process, you should be able to say, okay, well, I have learned that, and I tick it off, and just go through the list like that. What that would help you do is that at the end of every topic, you'll be able to figure out which topics you haven't covered or which sections you just don't understand, and you then know how to target your self-practice, or even how to ask for help that is targeted at what you really need to know. So yeah, that's it from me for this video. I hope you found it helpful. I wish you guys good luck. I hope you are enjoying the summer holiday, and I will soon start posting videos of past papers for the October-November guys. So please, if you haven't hit the subscribe button, please make sure you do so, and invite your friends to join as well. Have a good time, everyone. Until the next video, goodbye.